Hello guys, good morning. In this lecture today, I'm going to talk to you about G protein coupled receptors. The overview of today's lecture will be the introduction of the G protein coupled receptors and also the structure of G protein coupled receptors and how the signaling occurs via G protein coupled receptors, but my emphasis will be on CAMP pathway. And finally, I'm going to ask you a question which will test your knowledge of the lecture. Okay, so this is going to be the overview of today's lecture. So now coming to the introduction. So G protein coupled receptors. G protein coupled receptors are also called G protein linked second messengers or G protein coupled receptors. You might be wondering what does this letter G stand for? The letter G it stands for guanine. So basically G protein is guanine and the G protein stands for guanine nucleotide binding proteins. Okay, so G stands for guanine and G proteins are guanine nucleotide binding proteins. So what are these guanine nucleotides that these G protein bind with? So one is guanosine triphosphate or GTP, guanosine triphosphate, and the other one is guanosine diphosphate or GDP. The difference between GTP and the GDP is the phosphate group. In GDP, as the name suggests, we have two phosphate groups, and in GTP, as the name suggests, we have three phosphate groups. Okay, so this is the difference. Why these G protein coupled receptors are called G protein coupled receptors? Because they are associated with G protein, and this G protein is guanine nucleotide binding proteins. Okay, so the introduction is G proteins coupled receptors. These receptors are physiologically very important because they are involved actually in the regulation of sense of smell, sense of taste, behavior, and also the regulation of immune system. And some G protein coupled receptors are also involved in growth. Okay, that's why they are very important. They are involved physiologically in a lot of activities. So they are only found in eukaryotes. You must remember this. These G protein coupled receptors, they are only found in eukaryotes. They are not found in prokaryotes like bacteria. And the G proteins are the largest known class of membrane receptors. So they are the largest known class of membrane receptors, and they are also very diverse. And with the G protein coupled receptors, a large number of ligands they can bind with the G protein coupled receptors, and these ligands can be hormones, neurotransmitters, light sensitive compounds, etc. These G protein coupled receptors they are the largest known class of membrane receptors, and they are also very diverse. They are the target of 30 to 50% of modern therapeutic drugs, which means that the 30 to 50% of the drugs that we take in these modern age, actually these drugs target G protein coupled receptors. Hence they are very important receptors to study. And also another important thing that I want to add here is that many G protein coupled receptors, they have unknown functions, and these functions are yet to be determined. And in fact, the Nobel Prize in Chemistry in 2012 was awarded to the work in G protein coupled receptors. Okay, so from this introduction, what we can say is that the G protein coupled receptors are the largest known class of membrane receptors, and they are actually the target of 30 to 50% of modern therapeutic drugs, and they are found only in eukaryotes. And also they are very important physiologically, which are involved in regulating a large number of activities such as sense of smell, taste, behavior, immune system, and growth. So now coming to the structure. So what is the structure of the G protein? So now I want you to have look here. So the structure of the G protein consists of 7 alpha helices. If you count 1, 2, 3, 4, 5, 6, 7 alpha helices. Transmembrane 7 alpha helices. So this is the outside extracellular part, that means outside of the cell. This is the inside of the cell. So these 7 alpha helices, they come from extracellular to the intracellular. So the structure of the GPCR is consists of 7 alpha helices. Okay, this is 7 transmembrane alpha helices, right? These are called alpha helices and they are transmembrane. Okay, this is the most important point. And because these G protein coupled receptors have 7 transmembrane helices, 1, 2, 3, 4, 5, 6, 7, hence they are also called the 7 transmembrane receptors. Okay, G protein coupled receptors, they are also called 7 transmembrane receptors. And as the name implies, G protein coupled receptors are associated with G protein. Okay, so what this is the part of the G protein coupled receptor, and this structure here is G protein. Okay, this is G protein. So this 7 alpha helices, that is GPCR, is associated with G protein, and this G protein actually they are heterotrimeric, meaning that they have 3 different subunits. You see here 1, 2, 3 subunits, alpha subunits, beta subunits, and gamma subunits. So G protein coupled receptors are associated with G protein, and this G protein has heterotrimeric structure, that means it has 3 different subunits, alpha, beta, and gamma. Okay, so now what's more, if you look carefully here, this alpha subunit and the beta subunits, they are attached to the cell membrane. This part here this is the part of the cell membrane. Alpha and beta subunits are attached to the cell membrane by lipid anchors, whereas gamma subunit is attached to the beta subunit. Okay, so this is the structure of the G protein. Structure of the G protein consists of 7 transmembrane alpha helices which are associated with G protein, and this G protein is heterotrimeric. It has 3 subunits, alpha, beta, and gamma. Okay, so now the most important thing, how the signal transduction occurs via a G protein coupled receptor. This is what I'm going to discuss here. Okay, G protein coupled receptor in inactivated state, it is going to be like this. When there is no ligand bound, we have here G protein coupled receptor, and this is the G protein. And if you look carefully in the G protein, this alpha subunit is actually bound to GDP, guanosine diphosphate, in inactivated state. Okay, in inactivated state, G protein is bound to GDP. So this is the inactivated state. Why it is inactivated state? Because there is no ligand binding here. This site here is the ligand binding site, right? So then what happens when ligand binds? For example, this red thing here, this is the ligand. When this ligand binds to the G protein coupled receptor, and the ligand binding site of the G protein coupled receptor is extracellular, as you can see here. Then what happens is that the first important point is the binding of the ligand to the G protein coupled receptor, and after that this G protein coupled receptor, GPCR, it undergoes conformational changes. Okay, G protein coupled receptor undergoes conformational changes. Ligand binds to the G protein coupled receptor, and G protein coupled receptor undergoes conformational changes. Okay, and then what happens? So after it undergoes conformational changes, this alpha subunit actually exchanges GDP for GTP. So here in inactivated state, GTP was here and GDP was associated with alpha subunit, but in the activated state, alpha subunit will take GTP and it will give GDP. So basically alpha subunit exchanges GDP for GTP, right? So this is what happens when ligand binds to the GPCR and the GPCR undergoes conformational changes, right? And the next point is after this exchange, what happens is that this alpha subunit, actually here you can see that alpha subunit is bound with beta and gamma. So beta, with beta gamma dimer, but here it will dissociate. Alpha subunit dissociates from beta gamma dimer. Okay, so alpha subunit dissociates from beta gamma dimer. So point number 1, ligand binds to the G protein coupled receptor. G protein coupled receptor undergoes conformational changes, and alpha subunit exchanges GDP for GTP, and alpha subunit dissociates from beta gamma dimer. Here you can see that alpha subunit is associated with beta gamma dimer. Here it is the dissociation. Okay, and then what happens when this alpha subunit is dissociated? Then what this alpha subunit does is that it regulates target proteins, and target proteins relay signal via second messengers. So what happens? This alpha subunit regulates target protein. These target proteins can be ion channels, they can be different enzymes. So I would also like to add here that this beta gamma dimer, they can also regulate target proteins. They can also regulate target protein, but our focus will in this lecture today will only be on alpha subunits for this mechanism of G protein coupled receptor signaling. Okay, so this is what happens. And the next point is, okay, fine. So if when the ligand is bound, this alpha subunit is regulating target proteins, and target protein relay signal via second messengers, but it will keep on going there. Therefore, it has to stop. So how this pathway is stopped? This pathway is stopped when this GTP actually is hydrolyzed to GDP, and once this happens, everything goes back to normal, and this happens on its own. For example, how the regulation of this pathway occurs is that the hydrolysis of GTP occurs and everything goes back to normal, like here. Okay, from this scenario here, it goes back here. Okay, like in the inactivated state where the G protein coupled receptor can take one more ligand and the signaling can occur, right? So one way of regulation is the hydrolysis of GTP, and this happens on its own. Okay, and the other way of regulation is that our body has few other ways to regulate it. For example, regulation of G protein signaling via RGS protein, regulation of G protein signaling. So this pathway is actually brought back to the inactive state. Okay, G protein, like this state here, from this to this. Okay, so fine. So what here I discussed like how the signaling occurs via G protein coupled receptors. So what are the different steps? First step is that ligand binds to the G protein coupled receptor. G protein coupled receptor undergoes conformational changes, and after it undergoes conformation changes, then alpha subunits, it actually exchanges GDP for GTP, like here, and then alpha subunit dissociates from beta gamma dimer, and alpha subunit actually regulates target proteins, and target proteins actually relay signal via second messengers. And the regulation of this pathway, the control of this pathway is by the hydrolysis of GTP and also by different regulatory proteins such as RGS, regulation of G protein signaling. So now I want to give you an example here to show you actually how this G protein coupled receptor is activated upon ligand binding and how the signaling occurs. So I will take the example of epinephrine. So now I want you guys to have a look here in this figure. So let's say that the ligand is epinephrine. Okay, ligand is epinephrine. So this structure here is the G protein coupled receptor. Okay, fine. So when epinephrine binds to the G protein coupled receptor, what happens is that the G protein coupled receptor undergoes conformational changes. So the G protein coupled receptor for epinephrine will be adrenergic receptor. Okay, adrenergic receptor, and epinephrine is involved in fight or flight response. So epinephrine is involved in fight or flight response. So when epinephrine binds to the G protein coupled receptor undergoes conformational change, and alpha subunit exchanges GDP for GTP. So here you see GDP, but alpha subunit exchanges GDP for GTP, like here, ligand bound, and alpha subunit conformation change, and alpha subunit exchanging GDP for GTP. And then what will happen is that now alpha subunit dissociates from beta gamma dimer, yes, and then what it will do, then alpha subunit will regulate target protein. Alpha subunit will regulate target protein, and in this case the target protein is an enzyme that is adenylase cyclase. Okay, so what alpha subunit does, it regulates adenylate cyclase enzyme. The function of adenylate cyclase is that it converts ATP to CAMP. Okay, it converts ATP to CAMP. This is the second messenger, right? So now when this CAMP level is high, then what happens? Actually, it increases heart rate, it dilates blood vessels, and it also converts glycogen to glucose. Fine. So the example is epinephrine binds to G protein coupled receptor, like here, and G protein coupled receptor undergoes conformational changes. After it undergoes conformation changes, alpha subunit exchanges GDP for GTP, and alpha subunit regulates adenylate cyclase, and adenylate cyclase actually converts ATP to CAMP, that is cyclic adenosine monophosphate, and which is a second messenger, and it causes actually increase in heart rate, dilated blood vessels, and conversion of glycogen to glucose. Okay, so this is the mechanism of how signaling occurs via G protein coupled receptors. So now my question to you is that why CAMP is called second messenger? Okay, so why CAMP, this cyclic adenosine monophosphate, why it is called second messenger? So please guys, please answer this question. Please comment below and answer this question, and whosoever answers it correctly, one of the individuals will get the prize from our channel. Thank you very much.